Okay, so welcome all of you. We are starting today the second edition of the Liver Seminar Program. As you know, is an educational program funded by Gilead Sciences, the CIBER-EACD, the CIBERET, and the Asociación Española para el Estudio del Hígado. And this year, again, we have 9 excellent speakers coming once per month to Barcelona or Madrid and having the broadcast, the live broadcast between the two institutions. Today we start with Professor Krista Rombout, but we'll have Stephen Dooley, we have Robert Porte, we have Jonel Trevica, we have Deborah Barks. We have so many people coming to Madrid or Barcelona during this year, so I hope you take advantage of that. Everything is already, most of the speakers are already confirmed at the website, www.liverseminars.eu. And I think we can start with today's seminar. Today we have the pleasure, I think we have enjoyed so far the day with Krista Rombout, and I hope that those groups that have met with her also have enjoyed her expertise. Krista Rombout, as you know, is a professor at the Regenerative Medicine and Fibrosis Unit at the University College of London. She holds a PhD in medicine that was from the Free University of Brussels. And as you know, because she is one of the most important experts in fibrosis and hepatic stellate cells, she did the postdoc and stayed for some years at the pathology lab at the University of Florence. After that, in 2012, she moved to London to start there a new avenue, obviously with the same research interests that, as you know, are fibrosis, hepatic stellate cells biology, cancer cell biology, NAFLD, NASH, 2D, 3D cultures. I'm sure we'll have a mix, a potpourri of everything in this presentation. And as, just two numbers, Professor Rombout has more than 50 peer-reviewed publications in top-rated journals. She's a great mentor. She has supervised so many PhD students, master students, and bachelor students, and also is editorial board member of different journals, nowadays including Journal of Hepatology and American Journal of Physiology. So with that, I want to personally thank you for being the first speaker of the second edition of the Liver Seminars program, and we look forward for your presentation. Thank you, Krista.
Hello, everybody. Jordi, thank you very much for inviting me in the nice presentation. I hope I can do a very nice presentation for you as well. I'm very honored to be here, and I would say hello, Madrid. So today, what I am going to talk about is I'm going to try to invite you to understand how we do believe, and many other peoples, we believe how we need to work with the hepatic stellate cells. So just for honesty, these are my disclosures. I'm going to give you an overview of the hepatic stellate cells in liver fibrosis. Antifibrogenic compounds. We are desperately needing the antifibrogenic compounds. And where do we start and what is the strategy? Of course, we know we have cell lines, we have primary cells, we are working in 2D, 3D animal models. So I'm going to give you a little bit of flavor of all the things that we can do with. Stellate cells, of course, very important in human liver disease. We do have the stimuli and the mechanisms, and we don't know yet all of them. And of course, also a few examples of hepatic stellate cells in NAFLD, NASH, and in tumor development. So as antifibrogenic strategies, there are a few key issues and concepts that we do need to realize. So hepatic fibrosis, already a lot of you know that it develops with the different morphological and spatial patterns. So the process involves resident cells, but also distant cells. Different molecular mechanisms can underline the fibrotic process, of course, in those different settings. And the origin of the fibrogenic cells, therefore, can be partially very diverse. As we all know, fibrosis can develop from a portal side towards the central side, depending on the etiology, and also from the pericentral side towards the portal side. Again, different etiology can cause this. Of course, as the end result, we do have a progressive sinusoidal arterialization, and finally, of course, the cirrhosis. Very important, of course, already mentioned, different etiologies. So in the liver field, and especially the liver fibrosis field, for many years there were doubts. Do we need to find for every different liver fibrosis originated from different etiology, do we need to have a different antifibrogenic compound to target maybe different myofibroblasts? So this meaning that do we need different drugs for the different etiology-induced chronic liver diseases? Of course, over the years, in the beginning, let's say a few decades ago, when I started as a PhD student, there was only the hepatic stellate cell. So it was kind of very easy, very straightforward, we thought at that time, to target just that cell. If we know the cell, if we know how it reacts during liver fibrosis and progress towards cirrhosis, if we do understand that cell, we do find our compounds. Of course, this is not true, because lately portal fibroblasts have been shown to also become myofibroblasts or activated fibroblasts and produce the extracellular matrix. There was a time where it was also suggested that hepatocytes and cholangiocytes, they could move and transform due to an epithelial to mesenchymal transition into a myofibroblast. I think at this stage we already do know that this might be not the case, but anyway, the question mark is still there. And then of course the cells from the bone marrow. We do know that stem cells as well as the circulating fibrocytes, they are moving towards the injured liver and they will become the myofibroblast. Of course, for all these cells, quite a bit of different roles have been established or suggested as the pro-fibrogenic, pro-angiogenic, modulating the immune system and in liver regeneration. Of course, very important, because as I mentioned a few years ago, there was the doubt like how important the hepatic stellate cell was in liver fibrosis process. But then again, very nice paper published by Robert Schwabe, showing that by tracing the stellate cells, and he showed that stellate cells are dominant contributors in liver fibrosis and independent of etiology. So actually, for somebody who had been working already for many years in the hepatic stellate cell field, I was actually very glad that this was confirmed another time with very elegant methods. What do we have so far in the clinic or in clinical trials to target the hepatic stellate cell activation? Yeah? So here you can see a few of them, phase 2 or phase 3 trials. I'm not going to discuss this, but what you can see, it's or agonists of receptors. It can be a ligand, it can be a neutralizing monoclonal antibody. So let's hope that maybe one of them can reach and go into the clinic. Of course, very important, and this is not just for the liver fibrosis field or for the hepatic stellate cell in general. This is a very nice review paper. The reason for failure in clinical trials, the main reason for failure is not the safety, but it's the lack of efficacy. And I will come back to that one again. So the hepatic stellate cells, where do you start? Because of course, if you start new in the field, stellate cells, they seem kind like of myofibroblast. And the more we learn about the cell type from the quiescent phenotype or the activated phenotype, of course, it's known to be a very complex cell, as many cells, of course, in other diseases. So a lot of research has been defined. Different signaling pathways, different molecules that we could target, going from the plasma membrane towards the cytosol, and of course also in the nucleus, interfering with the transcription. This is a typical slide that I show where we need to take care of, to think about once in a while, right? So, of course, when you work in 2D or in your cell culture, the findings that you have, they might not reflect of animal models or might not be reflected in the human chronic liver diseases. So here you already can see that these are human hepatic stellate cells. They are transforming from a quiescent phenotype towards an activated phenotype on 2D in plastic. Of course, a lot of us, we have been trying to find a compound to inhibit the quiescent, the activated hepatic stellate cell, to push it backwards to the quiescent phenotype. But once we started to work in vivo in an animal model, it was not success. Yeah? So bear in mind, always try to find you the right animal model. Of course, animal models, in most of the models, cirrhosis develops within 4 to 8 weeks. Here is an example of the CCL4 injection. So already different models do exist using the CCL4. And of course, no model is identical or even similar to the chronic liver disease in human. What is very important, again, for the animal models is that you really need to think before you start working with animal models. And I saw this morning very nice examples of the correct animal models used to investigate a certain topic. This is, for instance, is a paper of the group of Florence of Fabio Marra, where he does shows that the CCL2 ligand, very important in liver fibrosis development and hepatic stellate cells, it affects in a different way inflammation and fibrosis. And just because you're using two different mouse, different species, yeah. So Black 6, BALPC, very important. Moreover, this is a more recent paper where somebody has been investigating, an American group has been investigating that actually the NASH models that we are using at the moment with our mice grown into the facility, the animal facility, they are absolutely not anymore showing us how actually we are as human beings. So the species that we are using, whether it's a BALPC or a Black 6 mice, it doesn't represent the human disease. Very important, she has been using wild mouse, so they went really chasing mice in parks and so on and so on. It's very beautiful paper, very interesting. The Western dye that we're all using in the lab is not working on these mice, so they are now trying to investigate a little bit better. What is very important in most of the animal models is that when you remove the insult that activates the hepatic stellate cell and to go into liver fibrosis, cirrhosis, if you remove the insult, then automatically these hepatic stellate cells, activated hepatic stellate cells, they become deactivated or inactivated. And this is more or less also something that we see in the clinic. When a patient stops taking strong medicine, alcohol abuse, and so on, we do see that liver fibrosis is regressing. So very important, there are models, and also what I do know now from Jordi's group and the other groups here, they already are very much investigating this. So one of the examples that what we can do with human hepatic stellate cells is working with the cytoglobin B. Cytoglobin B is more or less the only stellate cell marker for the human hepatic stellate cells. We do know, of course, that stellate cells, human hepatic stellate cells, when they become activated, they are expressing the alpha smooth muscle actin, but this is a myofibroblast marker, and we do have plenty of different fibroblasts from different origins. So very important for the human hepatic stellate cell field is the cytoglobin B, and this is in collaboration with Norifumi Kawada from the Osaka University, where he shows that FGF2 is very important to keep the cytoglobin B upregulated to have a quiescent hepatic stellate cell. Besides the in vitro data, they also performed the in vivo data, showing that indeed if you treat the CCL4 treated mice with FGF2, you get a regression of the liver fibrosis. So this will be presented during the AASLD meeting. One of course, one very strong stimulator of liver fibrosis process and of the hepatic stellate cell is the oxidative stress, of course. And oxidative stress is promoting the disease progression by, of course, promoting the hepatocyte injury, the inflammation and the activation of hepatic stellate cells, and therefore the extracellular matrix production. So here, this is Lisa Longata. She started as a postdoc fellow a few years ago. What she has been unraveling, she has identified a new ROS species. What is very important in comparison to the already known effectors of the ROS species, the reactive, the gamma ketoaldehydes, they are promoting the activation of the primary human hepatic stellate cells in vitro. They are promoting them as low as 500 picomolar, so that is extremely low, and with the other ones that we do know already, they are like micromolars working. Moreover, what is very important, what we thought that oxidative stress automatically would induce the synthesis of extracellular matrix, so the profibrogenic stimuli, the gamma ketoaldehydes, they are not inducing the production of extracellular matrix, but they are more promoting the pro-inflammatory response and also autophagy, what she shows in the paper. Now, what about mechanotransduction? Very important for the hepatic stellate cells. As everybody knows, human hepatic stellate cells, or also rat hepatic stellate cells, they are migrating, they becoming very contractile, and they're migrating upon chemo gradients such as platelet-derived growth factor, the fetal bovine serum. Cells are migrating towards that. Now, this is Darius. He's one of my PhD students, which is working at the Imperial College Bioengineering. And actually what he has shown here is a technique that we don't know that much of in the liver field. It's a durotaxis. Durotaxis is measuring the migration or the effect of the migration by changing the stiffness of the substrate. And so what he developed was a durotaxis assay, what you can see here. These are, for instance, in one petri dish, you create a system where you have two different stiffness substrate, the same substrate, but with two different stiffness. So actually what happens, and this is also something that we do see in human liver diseases, the quiescent hepatic stellate cell will always migrate from the soft tissue towards the hard substrate stiffness. So of course, once that was established, Derek was very much interested in the app, and we do know that is also very important in human hepatic stellate cell activation. And what he shows is that actually it is the focal adhesion kinase. Can't find the mice. Okay, the focal adhesion kinase at the site where the cell is moving towards the hard stiff substrate. The focal adhesion kinase is connected with the YAP, and the YAP is performing the activation of the stellate cell and is improving the migration. So Darius has been working with all kinds of knock-in, knock-out, silencing, and the chemical inhibitors. Of course, 2D, working in 2D, it's kind of very straightforward because you don't need that much. You just need your plasticware and you need your cells. But of course, during the last decade, we do know that, of course, working in 2D, you are forgetting that cells normally are isolated from out of a 3D microenvironment. So here are a little bit the examples of what actually is missing in the 2D versus the 3D. Of course, also in our lab in London, we started working and optimizing a new 3D model. This is Giuseppe. He started as a PhD. He developed, starting from a whole human liver, he decellularized that liver. By saying decellularization, we have been removing the cells, and we were left over with only the extracellular matrix and the bioactive cues binding to the extracellular matrix. So what you can see here is, or you can go for the whole liver decellularization, or you can section small pieces of the liver in very small cubes, which then you can work on decellularize and can work in a 96 well up to a 48 well. You can repopulate these cubes again with all cells that you can imagine, the cells that you're interested in. So to show you the difference, and what is very important to show you the difference, is that this is a healthy liver scaffold, a SEM of a healthy liver scaffold, showing that the hepatocyte pocket here and surrounded, so normally here you would find an hepatocyte, and this is the extracellular matrix, a very thin band of extracellular matrix. Now when you decellularize a cirrhotic scaffold, then this is how it looks like. So this is the hepatocyte pocket again, but here you do see a thick rim of extracellular matrix. It's not only the quantity of the extracellular matrix that has changed, it's also qualitatively. So the cirrhotic, what we do know from a large proteomic study, the extracellular matrix of cirrhotic livers contains very unique and specific proteins, extracellular matrix proteins, and it's not only the collagens, what we all think. Yeah. So this is Elisabetta. She just started her PhD, and she has, during her master thesis, she has been investigating the effect, of course, of the primary human hepatic stellate cells between a 2D culture here, the gray bars, versus the 3D healthy liver scaffold. And then she has been treating the cells with TGF-beta, PDGF, and LPS, so common stimuli for the stellate cell activation. And already you can see that there are drastically differences between culturing the same cell of the same donor in parallel at the same time between the 2D and the 3D in the gene expression. What for us is very important is that she found that the same cells grown on healthy scaffolds, they are behaving differently on 3D cirrhotic scaffolds, what you can see here. Here the cirrhotic scaffold, for instance, you do have a huge expression of collagen type 1, and this is only by culturing the cells on a cirrhotic liver scaffold. So of course, for us, this opens the opportunity to search again for new targets in those hepatic stellate cells grown on liver scaffolds, the healthy versus the cirrhotic. Of course, not only we are making scaffolds. This is Luca. He's a PhD student. So what he started to optimize was, after the decellularization of the liver, he made a powder. So he lyophilized the liver powder, the extracellular matrix. He can make a solution again, or gellify it again, or as well we can 3D bioprint this gel. So what we actually do need is, we have the 3D printer. We are using an ink, a bio ink, which the company is giving us. Most of the liver cells are not happy growing cells in the bio ink of the company. But what happens is, if we dilute this bio ink and we are adding the extracellular matrix of the human liver, so that solution here, we add the cells, all cells are growing. So that's a good improvement of the 3D bioprinter anyway. So this is an example of the 3D bioprinted primary human hepatic stellate cells after 12 days, live dead staining the control cells with the TGF-beta treated cells. Very important here is again that if you go and extract the cells out of the gels again, what you do see here, for instance, is that the 3D biogel derived from the human liver is favoring the quiescent phenotype over the phenotype that you can find here in the 2D. So definitely these are two, this is again a nice model of how to investigate the differences in activation and quiescent state. So this 3D bioprinted material with the primary hepatic stellate cells, we continue further for high throughput screening. Another model where I have been working on in collaboration with two companies in London, or in the UK anyway, is the liver chip, and this is what you see here in the incubator. So very nice, everything is under control, and very important here is that each liver chip is independently controlled and it's a dynamic system. So the liver chip was already on the market by the company with primary human hepatocytes, but what we wanted to do, we wanted to create a NAFLD/NASH triple cell culture. So we have been working and optimizing the triple cultures on the liver chip with primary human hepatocytes, the hepatic stellate cells, and primary human Kupffer cells, as you can see here. Very important, of course, the optimization took quite some time, as of course every cell needs a very specific medium. So before we understood which medium to be used to have happy growing cells in general, whether it was the hepatocytes, the Kupffer cells, or the stellate cells, it took some time. But anyway, we succeeded. We also have been treating the cells with oleic acid as a stimuli for the NAFLD/NASH development, as well as SPS, LPS for the pro-inflammatory stimuli, the Kupffer cells and stellate cells. We have been analyzing all kinds of things, phenotypic analysis, the P50 activity, proteomic analysis, as well as the transcriptomic analysis. Of course, at that time, a lot of papers popped up about the patatin-like SNP mutation that we find in human livers, and which shows to be very important in speeding up the process, whether it's NAFLD/NASH and whether it's cancer development. So, of course, one paper already showed very well that the patatin-like SNP mutation in the primary human hepatic stellate cell is actually more pro-fibrogenic and pro-inflammatory. Of course, what we did as well, we have been genotyping all the stellate cell donors that we have, and what you can see here, an allelic discrimination genotyping we performed. So it's a little bit more than a qPCR, a single qPCR, but it's very straightforward technique. What you do see here are 3 patients, stellate cells from 3 patients, A, B, C. And after the genotyping, we do have the wild type for patatin-like, we have the heterozygous mutant, and we have the homozygous mutant. Very important is that these 3 genotypes, they are behaving differently, whether it is you are investigating this in 2D on plastic or in 3D cultures. Very important. Of course, in our triple culture for the NAFLD/NASH model, what we inserted was this. Is the patatin-like SNP mutation so important? So actually, we set up the triple culture again, but the only variation here was using the patatin-like mutants. These are 3 donors, yeah, versus the wild types, and then adding the hepatocytes and the Kupffer cells, yeah. We have been, this is one of the examples of the Luminex, a 26 analyte Luminex was performed, and this is as a typical example, the interleukin 6 in the medium measured after 8 days of culture and 15 days of culture. What you can see already is that those triple cultures where you do have the patatin-like mutant, they are already very strongly primed, whether it's with the oleic acid treatment or the oleic acid with the LPS. So definitely they are different. Now, what about not the direct crosstalk of different cells, hepatic cells, with the hepatic stellate cells, but from a distance? So this is Zhen Zhang. She was our postdoc for 2 years, and what she actually investigated was the effect of the adipocyte visceral tissue. So from the distant, the conditioned medium of those cells on hepatic stellate cells, again using the 3D healthy liver scaffolds. Yeah. So in collaboration with Schalk van der Werme of the University of Leuven, Belgium. So they had been stratifying their patients. During the bariatric surgery, they have been taking the adipose visceral tissue, put it in culture. The conditioned medium was taken and sent to us, and then has been identifying certain effects, and this is the design of the study. What you can see here, which is very important, so here we do have the different conditioned medium of the different patients, and what you can see is that the visceral adipose tissue conditioned medium of diabetic patients, very much to think about when you start performing these experiments, because they are inducing a much more robust effect on the pro-fibrogenic activity of the primary human hepatic stellate cells versus the visceral adipose tissue conditioned medium of the non-diabetic patients. In collaboration with another group from Oxford, we have been doing the same kind of experiment, but we have been treating the primary human hepatic stellate cells with the serum obtained from bariatric surgery before and 6 months, 12 months later. And there again you do see that things in the serum of those patients is drastically changing, and therefore also the human hepatic stellate cells pro-fibrogenic action or the activation state. Now, what about the hepatic stellate cell tumor stromal interaction? So we already know for a very long time that hepatic stellate cells and the cancer cells, they are cross-talking one another, right? So, or it's the cancer cells are cross-talking with the hepatic stellate cells, and hepatic stellate cells are becoming more activated, or differently, more papers are shown that hepatic stellate cells are cross-talking with the cancer. Moreover, what is also important is how the energy mechanism, as well as the pH sensors in those cancer cells or stellate cells, are changing upon the tumor formation. So just to start here, I will give briefly an overview of experiments that we have been performing on two sensors. Very important: the AMPK, or the adenosine monophosphate activated kinase, which is a regulator of the cell metabolism. It's an energy sensor, so this AMPK molecule is detecting the ATP. If a cell has a little bit too less ATP, then the AMPK will provide and shut down all processes that the cell doesn't need to increase again the ATP levels. On the other hand, we do have the VATPase. It's a proton pump at the plasma membrane, and it's a key regulator of intracellular pH. Again, due to the fact that most of it is found on the plasma membrane, this would automatically indicate that this the intracellular pH changes are picked up and sensitized by this molecule, and then it will shuffle and expel the protons to get a better pH inside the cell. Now, in the cancer development, this is very important. AMPK can be a tumor suppressor, or it can function as a tumor promoter. So again, here, and I'll show you later, it's kind of very complex system. The VATPase actually is important as a pro-oncogenic signaling for tissue invasion and metastasis, cell survival, of course, because it's regulating the pH, it's adjusting the cell to adjust to the pH that it feels inside and outside, as well as drug resistance. So in a previous study here, we had a large cohort of patients, and with this protein, the detection of this protein was differently in the different stages of cancer development. So at one point, when I became interested in the AMPK, I asked to perform the AMPK alpha 1 subunit to perform the immunohistochemistry. And actually, this is just one patient, and it didn't matter how many patients you go and check the healthy tissue versus the cancer nodule. There was a there was a lot of confusion. We couldn't find some kind of a strategy. What is very important, of course, we have been going to the TCGA, and indeed what you can see is that here the AMPK alpha 1 is not significantly up or down regulated depending on the staging of the patient. On the other hand, what we did find was that AMPK alpha 1 subunit is significantly upregulated in the protein 53 mutation of those cancer patients versus the catenin in the other patient. So this is Catherine. She's a PhD. And during her PhD, one of her projects was to start working on the paracrine effect of the hepatic stellate cells on HCC cells and vice versa. And as a first tool, we started to work on the crosstalk, see whether the cancer cells are dominant in crosstalking with the stellate cells. So there's a reason why we have been using these two, of course, as mentioned in the previous slide. The PLC5, they are characterized by 53 mutation and the catenin in HepG2. Of course, this is all further related to the human samples and the studies, the big omic studies, where they do show that you do have different subclasses of HCC in humans. So actually what Catherine did, she has been obtaining the conditioned medium of these two cell lines, and she has been culturing the human hepatic stellate cells. Of course, what is surprising that the human hepatic stellate cells, they are not reacting that much on the level of pro-fibrogenic genes, but much more as a pro-inflammatory stimuli. So actually what does this means that is the cancer cell, they need hepatic stellate cell to increase the pro-inflammatory signals in the cancer medium. Of course, very important to keep up the development and to get resistant for the immune system. Very important here is that if you put these both the conditioned medium of both cell lines on top of the hepatic stellate cell, this cell line here, the PLC5, is not interfering with the proliferation of the hepatic stellate cell, whereas the HepG2 cells, they do. Very interesting here. This is the phosphorylation of AMPK on a serine residue. Most of the time people do forget to investigate this as well, because the classical one is this here, the AMPK phosphorylation on the threonine 172. But what we did find out was that this effect here was basically because of this serine residue. It becomes phosphorylated in the hepatic stellate cell, and actually this phosphorylated AMPK is inhibiting the phosphorylation of the other AMPK. And therefore, these cancer cells, they can communicate with stellate cells and induce the proliferation of hepatic stellate cells. So here we have discovered this kind of two-way dual effect of on AMPK system. Of course, Catherine has been defining this system much better. She has been working with activators and inhibitors, and two different signaling pathways have been identified, and which are now again investigated for novel commercial available inhibitors. On the other hand, this is the work of Juicy. Juicy has been working on the VATPA, so the proton pump on your membrane. So as you can see, it's quite a complex proton. Not all of the subunits are as important as a few of them. We started all with the VATPAs, besides my interest in coupling the VATPAs with the AMPK, because I thought there might be a link in stellate cells. Also on a regular basis, there are quite some publications showing that these pH regulators, they are very important in tumor development. So actually what Juicy did is she has been first, of course, identifying the VATPA subunits on the primary human hepatic stellate cells, in the activation of the cells in culture, and also in the normal and diseased liver. Of course, very important it was to find out whether indeed the pH has been changed when you inhibit the VATPase pump. But what was very important is that if you inhibit the proton pump, you activate the AMPK. So both these systems, they are connected inside the primary human hepatic stellate cell. Moreover, what she also has been doing the reversed way, if you pharmacologically activate the AMPK, you downregulate the VATPase expression. So again, proving that you have a full circle in the system in the primary hepatic stellate cell. And very important, and maybe in contrast with the stellate cells from rat and mice, in primary human hepatic stellate cell, AMPK alpha 2 subunit is not that much expressed, so it's the alpha 1 that has a key regulator regulation in the V8PAs in human hepatic stellate cells. Of course, a BDL model, so the in vivo model, we have been verifying that as well as, of course, as always, you need to go back to the human tissue in healthy and chronic disease liver versus the alpha smooth muscle actin and the V8PAs. And there we did show indeed that the V8PAs is highly upregulated. So in conclusion, I hope I could persuade you to believe that hepatic stellate cells, they are a key target in liver fibrosis. For the antifibrogenic strategies, we definitely started. When I started my PhD, there was nothing on the market, so that is that's nice news actually for me and also for the next generation. We are already started working on that one. Very important is that we need to have a mix of 2D models and 3D models and different animal models, yeah, to identify. So actually here, the more the merrier. Yeah. We still need to define much better the stimuli and the mechanisms of the hepatic stellate cells. And of course, I hope I could convince you that hepatic stellate cells, they are important in NAFLD-NASH process, as well as in the tumor initiation and in progression. So these are the fellows of the last 5 years in London. And actually, to tell you the truth, there's only one UK-based born person. Yeah, and all the rest we are coming from everywhere in the world. So it's a very national, international bunch of people, very motivated, very driven people. And these are a few of the collaborators. I thank you for your attention, and I'm happy to answer your questions. Thank you for your question. Actually, we are also performing experiments with the primary human hepatic stellate cells on the healthy liver scaffold, so the 3D model, healthy versus cirrhotic. And indeed, we are looking at NOx, NOx4, and oxidative stress, yes. And it's already due to the fact that TGF-beta is involved. Right, that we do know TGF-beta, we can't target TGF-beta because it's too big. It's more the medusa, right? It can harm, but it's also very good for your cells. So in liver pathology, and then especially with hepatic stellate cells, we do know that we can't target that. But indeed, I do believe that both those studies, very interesting, very important, and yeah, I do believe that oxidative stress, very important from the point of view instead of only targeting the primary human hepatic stellate cell, it's more like trying to prevent a pro-inflammatory reaction, yeah, or protect the stellate cells from a coming in pro-inflammatory insult, as well as the oxidative stress. That I think is the future to get back a deactivation of the stellate cells in the models. That's of course a very important question, and two big differences in using HUVEC cells. Yeah, most of the time they are homing back where normally the vessel wall was. Normally, sometimes it doesn't work, and with the cirrhotic scaffold we have a little bit more difficulty in getting them back where they should be. Now, human liver sinusoidal endothelial cells, besides trying to optimize the isolation method, we also have been going for commercial available endothelial cells, and there what we do see is that there is not this kind of homing response. But I must mention that what we did find was that most of those vials that we bought from a commercial provider, they were also contaminated with fibroblasts. So we can't rule out actually, is it because there were already the fibroblasts, they become activated, they are also moving into the scaffolds. But indeed, we very important. This is more important for the regeneration of the liver, because of course we have plenty of students, and one of the master students started to recellularize the human liver, the whole human liver, and there we definitely, excuse me, we need to have the endothelial cells before we can actually have something like a liver to recellularize with the hepatocytes, for instance. Yeah, so that is going to be a tricky point to solve. Yeah, indeed. Let's say from a human point of view, we do know that first you do have the hepatic stellate cells, because they are already in the place where the neoplasm is growing. Yeah? Later onwards, these cancer-associated fibroblasts are popping up. Yeah? And indeed, I do believe that due to the fact that they are phenotypically different, they are coming from a different fibroblast background, I do believe at one point that we need to take care and need to identify a possible second drug, because these are different cells. But yes, indeed, as well as the immune system inside your cancer is completely different. So indeed, we need to take that in consideration. Yeah. Although I must say that on the European Club in Bonn a few weeks ago, Robert Schwabe was giving a keynote lecture, and there popped up that of course the cancer-associated fibroblasts are coming later, but it seems that there were also hepatic stellate cells going into a cancer-associated fibroblast. Now, for the stellate cell field or for the liver field, this would be something new, a new concept, and I think we need to investigate that a little bit, because that I never saw in my cultures. The point is that in the pancreatic field, that is already established in the desmoplastic reaction, that pancreatic stellate cell can change into a CAF. But for the liver field, we don't know that. To clarify this, a few postdocs, they are working on co-culture systems. Yeah, so adding all kinds of different cell types together on the scaffold. The point is that we consider these extracellular matrix scaffolds something like a Lego puzzle, right? So you start with a few building blocks. It's not only the stiffness of the healthy scaffold versus the cirrhotic ECM scaffold that is important to drive the activation of stellate cells. It's also the bioactive cues that we find back after removing the cells. So we have data, but this is then for a project in the cancer field, is that if the cirrhotic liver scaffolds, they are full with bioactive cues such as the TGF-beta. So even if we remove the cells from the cirrhotic liver, the TGF-beta sticks there at the extracellular matrix and many other bioactive cues, meaning that it's not the stellate cells that are just changing because the stiffness is changing, it's also the signals that they get from the different extracellular matrix that is present in a scaffold. No? And your second question was
related to the shear stress.
The shear stress, yeah. So with the NAFLD-NASH, the triple culture, so that system we have been using, because it's a dynamic system, right? And we have been using this with our scaffolds, yeah. So, and we also get a very nice repopulation in the dynamic system. There is, for the shear stress, what you mentioned, at the moment we didn't check that yet, but we have been using the dynamic system for the primary hepatic stellate cells and the primary hepatocytes. And
You're at endothelial cells.
not yet the endothelial cells. Yeah, as I said, it's the endothelial cells, human endothelial cells. It's very difficult, first of all, to isolate. You know, you end up, at least I end up with a very low yield, unpure yield, and if you buy them from a company, they are contaminated with fibroblasts. So we are a little bit in a, we do have a small problem here.
Yes.
We have is, we have at the moment livers, human livers, of all kinds of etiology, but we are not investigating yet what age does on the extracellular matrix. And indeed, because you can imagine that although in the clinic the scoring F2, F3, F4 is already very well explained and everybody's using those guidelines. If you go and check on the extracellular matrix and for the decellularization process, it's quite different. No? So, of course, for every kind of etiology, we are trying to decellularize, to optimize the decellularization process and do the proteomics. But this takes time. Yeah, and then of course this is a very important question. Now I do know what aging does, and indeed that would be something very important to investigate, because I can imagine that a cirrhotic liver of a certain age has completely different ECM and maybe again other unique proteins that you can't find in a young cirrhotic liver or in a healthy liver. Yeah. A very good question, and I do believe at the moment in the stellate cell field, I do believe that at the moment nobody has very well investigated the possible differences between human hepatic stellate cells isolated from a liver, cirrhotic liver of alcohol-induced disease, or the NASH, the NAFLD, and so on. Indeed, already one of the reasons would be is because if you isolate, if you get the opportunity to isolate stellate cells from a cirrhotic liver, the protocol needs to be optimized a little bit better. The hepatic stellate cells, they are not exactly the same as from a more or less healthy liver, due to the fact that, of course, they all are also losing a little bit of their lipid droplets, which should be one of your very important factor to get them at the right gradient after your centrifugation, right? So indeed, I have been isolating hepatic stellate cells from NASH explants, so really the extreme one. I do see differences, yeah. Of course, those differences, I'm not sure whether this was because they are coming from the explant, so highly activated hepatic stellate cells, or because they were patatin-like mutated. On the other hand, the patatin-like mutation is not only related to those livers or those diseases that we know now, because I have been isolated quite different backgrounds actually, and you can find patatin-like in the most in all kinds of livers. The healthy livers, yeah, so the normal tissue where then you have, like for instance, the surgery for a cancer, there you also can find a patatin-like. So in London at the moment we are moving forward and we have optimized the program where we indeed are isolating stellate cells from different fibrotic background etiology, and then RNA sequencing and so on, and this gives beautiful data actually. So those few papers that arise with AMPK and tumor development in animal models, it's a little bit different, I think, from the human data that we have and from the human hepatic stellate cells. So what is known, of course, and then I'm going back to the clinic, people that have taken the metformin for so many years due to their diabetes, they have less chance of getting tumor development. Yeah, so that is pretty much a very good fact actually to start, and that was one of the reasons why I started to investigate the AMPK in the stellate cell tumor crosstalk actually. But indeed, you know, because there are a few papers on these animal models, and I must say that there's not that much of a good readout on these papers. So therefore, and as we are working with human material, you know, then it's kind of automatically that you just focus on your own work and just deciphering, defining again for another time to what happens in the human material actually. No. I don't think, because this was a big question in the stellate cell field. If we go for an antifibrotic therapy, do we need to target all the myofibroblasts, and why don't we induce apoptosis in all the activated fibroblasts, therefore also the activated hepatic stellate cells? Now they already have shown, but this is already many years ago, that actually this is not a good thing. Because of course, if you kill all the hepatic stellate cells, whether they are the good ones or the bad ones, you know, let's say your patient, which is then the animal, is not surviving very well. So actually, the antifibrotic compound should be inhibit the activation of the quiescent hepatic stellate cell towards the myofibroblast, or inhibit the myofibroblast to become more activated, but you need to get it pushed back towards a quiescent phenotype. But I don't think that there's a good guy actually.
Okay, so Krista, thanks so much. Wonderful presentation. Thank you. I'll see you, well, we'll see you tomorrow probably in the basic school, so thanks for coming to Barcelona. And the next liver seminar is November 28th from Madrid, alcoholic liver disease, hosted by Javier Cubero. Thank you for being here.
Thank you.